So really, before I begin, one question I want to ask all of you that attended the webinar today is, what is the impact of assay failure in your laboratory, and how do you monitor for it? Because that's in essence what we are looking to discuss and go into detail today between myself and Hadas, in particular on the focus of cell-free DNA and the application in liquid biopsies. So what you've got here is a very simple schematic of what is the point of liquid biopsies. You have your cancer patient, where you actually, what you do, you're taking blood or plasma from that patient. Alternatively, you could be taking urine. You're going through DNA extraction, then you're doing a diagnosis on that result. And the whole workflow for that is essentially driving better treatment for cancer patients. And the diagnosis could be leading to one of many routes. One could be triaging that patient for clinical trials. It could be a diagnostic marker where you're actually looking to see if there's any beneficial a reduction in the presence of a mutation following a specific treatment, or you could actually be using it as either a prognostic biomarker. Whatever you're using it for, you need to make sure that diagnosis or the diagnostic tool you're using at that endpoint is highly accurate and precise. The next, what I've done on the next couple of slides is actually break apart the workflow into two parts. The first part is pre-analytical, and this is very much where you'd be handling or processing your sample. So you're taking your blood or maybe your urine, you go through your DNA extraction step. You may have to actually transport your sample. That could be either pre or post extraction. There'll be a storage step, and then there'll be a quantification, and that could be done with a standard nanodrop or a fluorescence-based method. And all of this will then control and impact the quantity and quality of the DNA you have feeding into the analytical part of your assay. And in the analytical part, again, I've broken it down. It's a very simple workflow here. You've got your DNA out of your sample. Depending on the different type of platform that you want to work on, it will control the type of sample preparation you need to go through. So if you are going to be working in next-generation sequencing, there'll be a library prep step you'll need to go through. If you're working in digital PCR, you're likely to have to go through the droplet formation for that. And if you're looking at either running qPCR or even Sanger sequencing on these types of samples, you'll need an enrichment step to ensure you actually can detect the allele burden you're looking for at such a low level. And that will all then push into an actual decision. And the analytical side of the workflow is essentially going to be controlling the quality of the diagnostic result. And what's nice is when you actually plan out the workflow like that, you can see it's a very simple workflow. But what happens if it does go wrong, and how do you control for it? So what's that impact to you? And going back to the overview, at every single step of the process, you have different variables that can impact on the quality of the diagnostic test. So you have your sample collection, your handling. That could, depending on how you do that, it could increase the DNA fragmentation of your sample. It could even lead to contamination, which because you're going to be looking at, a lot of these biomarkers could be as low as 10 or even 100 copies, well, 100 or even 10 copies per 10 ml of plasma. If you have any contamination, you could be leading to false positives at the endpoint. You also have DNA recovery and DNA quantification. And the DNA recovery, is important to understand what the expected fragment size is from your sample. So if you're looking at extracting DNA from blood, you'll have fragment sizes of 170 base pairs, maybe a double nucleosome, so around 320 or 330 base pairs. And so you need to make sure you've got a kit that has a good efficiency for extracting DNA at a range of from maybe 120 base pairs all the way up to 400 base pairs. If, on the other hand, you're looking at urine, where the fragmentation size maybe is lower, the 70 or 80 base pairs, a kit that extracts at 100 base pairs or greater is not going to be very, is going to lead to you losing a lot of your DNA. And because the biomarker is so low in concentration in the plasma, essentially that would lead to a false negative result. And all those steps then feed into the final part where the DNA quality and quantity is absolutely critical. And that's where it will govern whether or not you end up with false negatives or false positives. False positives could be driven from a contamination from earlier in the process, and it's something to be very, very wary of. And also the quantity will certainly govern the specificity and sensitivity of your assay. I think that, for me, that's kind of the very brief introduction. What we are looking to do at Horizon Diagnostics is very much support the adoption and the development of cell-free DNA assays. Well, we've been working very hard over the last 9 months or 12 months in this area to develop reference standards. My colleague Hadas will be talking more about a couple of case studies we have, and then I'll round up with a couple of summary slides at the end. I'll pass over to Hadas, my colleague. Thank you.
Hi, hello everybody, and thank you for hearing us here today. So I'll talk you through about how do we actually manufacture reference standards, and then dive into 2 case studies highlighting the use of references to assess the sensitivity and specificity of cell-free DNA monitoring and different platforms, including next-gen and PCR-based methods as well. So how do we actually manufacture our reference standards? So at Horizon, we have developed a library of over 550 genetically defined cell lines, harboring mutations that are found in cancer patients, as well as we also generate the control cell lines that have the same genetic background. As part of our cell line validation process, as you can see in this slide, we have a highly validated process where we single-cell dilute all our cell lines to ensure clonality, and that's really important because we have pure populations of cell lines, and this is quite different than spiking in DNA from cancer cell lines that sometimes is being done in some laboratories. So just to make sure that you fully understand that our DNA is coming, or our reference standards are actually coming from very pure and validated cell lines from a common source. And then these cell lines are actually validated before undergoing engineering to produce the mutant cell lines. So before and after engineering, we run SNP6 array assays to validate the cell identity of the cell lines. Of course, we ran single sequencing on the mutant cell lines to assure that the engineering was properly done. And as you can see, we also perform digital PCR to measure the allelic frequencies and copy numbers of the mutants. And of course, RT-PCR to make sure that the gene is expressed. So using these genetically defined cell lines, we manufacture a range of reference standards, including DNA, RNA, FFPE blocks, and sections, cell slides for IFC and FISH. And now let's dive in more into the DNA reference standards. So as you can see here, there are actually 2 ways for us to provide you with the reference standards. One way is to actually give you the pre-diluted standard, so a mutant and a wild type, and then you can actually go through the process of making your own dilution And for example, here you can see one dilution curve that we have prepared ourselves for the V600K mutant and validated it using our digital PCR platform. Or alternatively, we can also support you by providing you with the mutants at low allelic frequencies, so following dilution. And dilution guidelines can be found on our website if you choose to do it by yourself. So, as I mentioned before, all our DNA products are going through extensive validation using our digital PCR assays. And if we do it in custom projects, we can get down to even 0.01%. Our controls have already demonstrated to have a profound effect on the labs that have adopted them. So we work all around the world with proficiency testing schemes, with technology developers, with the researchers and that are interested in a transition into the clinical use. And we're actually interested and continuously supporting researchers interested in comparing assay performance across platforms, validating their limit of detections of their assay, of their platforms or their assays. And of course, when setting up new assays, that's very crucial. So moving on to a couple of case studies. So now I would like to present to you two case studies. The first case study is a case study where we have produced an EGFR pentaplex. It's a reference standard with a range of allelic frequencies down to 0.05%. And this was done by a validation by digital PCR. Case study number 2, where we have also produced a multiplex cancer panel containing over 40 validated mutations. And I will also share with you some very interesting results we have been provided by one of our collaborators who has been using our multiplex. And I'll share with you the ion torrent data that was obtained by using our multiplex. So for case study number 1, as you can see here on the top left, we have 5 different mutants, 5 different EGFR SNPs and a deletion, that was diluted in 6 different formats, as you can see in the graph below. So we have 6 formats with different allelic frequencies, ranging from 2% down to 0.05%. And what you can see here is the digital PCR results of the total copy number of the mutant copies that were called in a total of 50,000 total EGFR copies. Now, how do we actually manufacture this kind of reference standards? So prior to blending, we analyzed all our singleplex ctDNA samples by digital PCR, and we determined the mutant and the total copy numbers per sample. Then based on these results, we then blend our DNA to form the different DNA blends that you see here. So once we establish the copy number with a high frequency mutant copies, then we go down and further blend our samples with a wild type to reach the very low allelic frequencies. So let me ask you a question. How good is your platform, if it's digital PCR or any other platform, in detecting mutations at 0.05%? Just take a second and think about it. And let's dive in some of the data that we've got. So now what I'm going to show you is only part of the data, of course. This is the data that we have obtained by digital PCR for our 6 samples out of the 6 formats. This is for the 0.05% blend. So this data was generated by digital PCR. And as you can see here, we have a list of the 5 mutants that were all blended in one tube, as well as the expected number of copies, around 25. So that's the expected number of mutant copies in a total of 50,000 EGFR wild-type copies. Also, to the right, you can see the observed mutant copies. And as I hope you can see, there's quite a good correlation between the expected and the observed mutant copies. And that's quite nice results when you consider when you consider how low down we can get down with the allelic frequencies. And another thing to consider is the wild type. So as you can see here on the right, we of course expect to get only 0 copies of mutant copies in a wild type sample. But as you will of course notice, this is not actually the case. We're getting some observed copy numbers, mutant copy numbers in the wild type. And this is actually, the results look actually pretty good for the first, third, and fourth assay. And this actually shows us how some assays are more challenging than others. Altogether, I think that this blend is pretty important, because we know that when we're talking about cell-free DNA monitoring, it is very important to be able to detect the low allelic frequencies. So this is very much relevant to the physiological and also to the clinical samples. So moving on to case study number 2. So for case study number 2, we have produced a multiplex reference standard we call TrueQ. And the way we have produced this multiplex is by combining many of our engineered cell lines, covering 40 mutations and more indigenous mutations. And that we did by blending actually 4 blends, as you can see here in pink, all the C blends, 1 to 4, each containing 10 mutations at 5% allelic frequency. These 4 blends were then combined together to form 2 unique V blends comprising 20 mutations at 2.5%, analyzed and quantified by digital PCR, and then mixed together again to form a unique A blend, as you can see here in green, comprising 40 mutations at 1.3%. Now, this blend was done with a defined copy number per microliter. We have 20 copies per microliter. And we can also dilute down this multiplex to 1 copy per microliter. So what is very interesting and useful about this reference standard is the fact that it recapitulates the complexity of tumor composition with over 40 validated mutations. So how good is your platform, your next-gen platform for detecting mutations at 1%? And one of our collaborators was actually asking this kind of question. And let me show you some of the data that we've received. So now I'm going to present to you some of the data that we have obtained internally, as well as some of the data that we have received from one of our partners on its Ion Torrent platform. Now, of course, the data set is very large, and just to get some flavor about the results that our partner was getting, let me talk you through some of I think we have here 8 to 10 different gene types we're looking at. So on the left panel, you can see the predicted allelic frequency that is expected for the 5% blend. So for the fact, and on the right panel, you can see the observed allelic frequencies that was observed using the digital PCR. And then to the right again, in blue, you can see our partners, our parent data. And what you can actually notice is that there is quite a good correlation between the expected and the observed data observed on the two platforms, the digital PCR and iron torrent, and that is for the 5% blend. Now let's continue with the 2.5% blend. Now you can also see that the results are pretty similar. When we use the 1.3% blend, then you can notice that most of the mutations are not called on the iron torrent platform. So that means that for this particular workflow, iron torrent could not call and is not sensitive enough to detect variants low as 1%. So I hope you can understand that this is a kind of experiment that is very easy to obtain results, giving you confidence about your platform, about your limit of detection. And also when you want to have a certain reference point when you're testing different platforms in your lab. So for anyone setting up their platform or optimizing assays, these are highly valuable reference standards to demonstrate how good you are at mutant calling and what's your limiting of detection. So some of our take-home messages and also some things we're actually very much excited about. So first of all, as Jonathan was saying, it is critical that cell-free DNA assays will be routinely validated to ensure accurate, precise, and robust reporting. So I hope I could convince you that highly validated reference standards are required to support assay monitoring and development. And finally, let me just tell you that we're very excited about launching a new type of product of reference standards specifically aiming for the cell-free DNA field, as we are developing reference standards that are not high molecular weight, but actually fragmented DNA. And that DNA actually recapitulates the DNA fragment that is obtained from plasma DNA. And of course, we are also interested in the urine reference standards as well. So that's it from my end. Let me.
So thank you, Hannah. That was a really good overview, and it's critical that you can see how the importance of adopting reference standards for both validating and also developing cell-free DNA assays. And I think what it always comes back to is, for you, what are now the outstanding questions? And if you take apart the question that we've been asking throughout the webinar, which is what is the impact of assay failure in your laboratory and how do you monitor for it, you can tease that apart to ask yourself, what sensitivity do you want to achieve with your assay? What mutations in particular are you looking at? And because of where we're positioned in being able to develop any mutations and mold that into any format that can support the adoption of cell-free DNA assays, how can we go about supporting the workflow? Now, based on the attendees of the webinar, I think it's then the next steps will be whether you're working in a molecular lab or whether you're in a next generation sequencing lab or a kit assay or platform developer. Please do contact me, and we'll be working together with Hannah to understand exactly what you're looking to achieve in the cell-free DNA space or arena, and we can generate reference standards for you. And so I think what I sort of bring this all around into a nice conclusion. We've had some great data presented by Hadas that shows the importance of reference materials in the adoption, development, and platform validation. And what we want to do now is really engage with you and support your work as we move forward.